Welcome back. In this video I'll explain quorum sensing in Vibrio fischeri. This is a required example in the updated IB biology syllabus. It comes under C2.1 cell signalling, which is a higher level topic. I'm going to include everything that you must know and some additional detail to provide context. So in the background we see the bacterium Vibrio fischeri and it's bioluminescing. The term used by the IB is quorum sensing. Notice also the terminology bioluminescence, a marine bacterium, cell signalling, and the species name. Now normally you're not expected to remember species names, however we haven't got a common name here, so I would suggest you actually use the term. So what is quorum sensing first of all? It's a type of signalling, it's used by bacteria and they use it to coordinate their behaviour based on their population density, i.e. how many individual bacteria are there collected as a group. The bacteria release signalling molecules, these are called autoinducers, and when a threshold concentration of autoinducer is reached, the bacteria will then produce this blue-green bioluminescent protein. Vibrio fischeri has a symbiotic relationship with this very cute Hawaiian bobtailed squid you see in the slide. So it's symbiosis, but specifically a mutualistic relationship. Now, for this case study, you don't have to know that this is a mutualistic relationship, but you later on in the syllabus you come across this kind of relationship, and it's a nice example and a nice link to make here. So, in a mutualistic relationship, both organisms benefit. So we see the bacterium, it receives nutrients from the squid as food. It also has a safe place to live, it's not free living in the water and ready to be eaten. The squid itself is a predator, it hunts at night, so by producing light in its light organ at night, it acts like an invisibility cloak. It's called counter-illumination, so it appears like the moon's light and it breaks up the silhouette of the squid when it's hunting, and it avoids it becoming prey to something else. So how does this bioluminescence work? Well, at a low population density, when there are a few Vibrio fischeri, they don't produce light. At high density, this autoinduces they accumulate, they trigger this light production. The binding of the autoinducer to its protein receptor triggers gene expression, and that results in a cascade of metabolic reactions within the bacterial cells, leading to bioluminescence. You don't need to know the details of that cascade, but sometimes understanding a little bit more does help. So here's what's really happening. The Vibrio fischeri are producing more autoinducer when they're at high density. This builds up and it will diffuse back into the cells of the bacteria. It will bind to the receptor protein, that's called LuxR. That forms a LuxR autoinducer complex. It then binds to a section of the DNA called the Lux operon, and that increases transcription of the genes involved in producing bioluminescent proteins, so it enhances gene expression. The enzyme luciferase is synthesized, and that will oxidize luciferin, producing or emitting this blue-green light. And obviously the bacteria are inside the squid, so therefore the squid's light organ will bioluminesce. So quorum sensing means you get a common response from all the bacteria present. They're all expressing genes for bioluminescence. Why do you need to know this? It's a case study that's linking to several IB concepts, so you have to know the case study for cell signalling, but you can also bring it into mutualism, you can also bring it into control of gene expression. So I hope you found this useful. If you did, please give it a thumbs up and subscribe so you can catch new videos directly targeting case studies expected by the new syllabus. The May 2025 exam showed that the IB really will test the details of the case studies they've included, so do make sure not to neglect this area. You've got this.